Chapter 13: Natural Substances and Supplements to Enhance the Cancer Immune Response.
Many cancer patients load up on numerous supplements to help treat their cancer. I certainly agree that many of these may be helpful. I have already discussed several within the other chapters of this book. There are an endless number of supplements, many with unknown benefits. In this chapter, I would like to discuss some of the more popular ones. I do not always recommend these to my patients, though that does not mean they may be without benefit. I hope to give an objective summary so that each patient can make their own decision if these are things they may want to add to their treatment protocol. Cannabis products. Certainly, you cannot delve too far into alternative and natural treatments for cancer without running into people saying that marijuana cures cancer. I have many patients who have tried cannabis products, but I have not personally run into any that were cured from using them. I wanted to know where these people who have been cured are. I attended some meetings where people discussing all the benefits of cannabis. The stories they described sounded promising, at least in the way they tell them to the lay public. But I did not see the real evidence. Many of the cancers they were citing great benefits for are ones that people can generally live with for many years, if not a decade or more. So, stating that a person was still alive after 1 year of cannabis use was not the result I was looking for. In regards to the immune response, research has shown that there are receptors in the immune system for compounds found in cannabis called cannabinoids. There are two receptors, CB1 and CB2. CB2 is found on immune cells. In an article published by Rader et al., May 20, 2009, in the Journal of Immunobiology called Cannabinoid-Induced Apoptosis in Immune Cells as a Pathway to Immunosuppression, they describe that cannabis inhibits the immune system and increases regulatory cells. These are not good things when it comes to cancer. So, how can there be all this information out on the internet that cannabis cures cancer, or at least helps? Certainly, it seems helpful with symptoms of cancer, but that does not mean it makes cancer better. In my research, I found that there was a generally overall lack of good studies on this topic. At the European Society of Medical Oncology, September 2017, Taha et al. Presented a poster titled, "The Effect of Cannabis Use on Tumor Response to Nivolumab in Patients with Advanced Malignancies." In their study, there were some concerning results related to cannabis use and cancer, showing patients using cannabis with the immunotherapy nivolumab had a greater than 50% reduction on their response rate, 37.5% versus 15.9%. On the positive side, there have been animal studies with CBD and a human study with THC directly injected into the tumors that have had some promising results. Also, cannabis is a complex mixture containing numerous compounds. It is very possible that specific isolated extracts may have benefits. We still have a lot to learn in regards to cannabis products and the treatment of cancer. Our group plans to conduct further studies looking at the immune response from intratumoral injection of cannabis extracts. However, it does seem clear that cannabis products can help with certain cancer-related symptoms, but this is more for palliation and not for actual treatment of the cancer. Silibinin Legacyl. Silibinin, which is derived from milk thistle, is used as a liver protector and is sold as Legacyl. A study published by Priego et al. Demonstrated that with 18 patients with lung cancer and brain metastasis who were given oral Legacyl, the response rate in the brain was 75%, with 20% demonstrating a complete response. The effect was more impressive in the brain than the body. Silibinin is an inhibitor of STAT3, which inhibits the immune response by increasing MDSCs and TREGs. STAT3 also promotes the secretion of TGFB, which, as we have discussed, is a potent suppressor of the anti-cancer immune response. 6-gingerol. 6-gingerol is the bioactive ingredient in raw ginger that inhibits arginase and helps convert M2, tumor-protecting macrophages, into M1, tumor-attacking phenotype. This is an important area of immune resistance and immune checkpoint failure. Also, Russo et al. Showed that macrophages might be an important mechanism that resulted in hyperprogression, rapid cancer growth, with the treatment of PD-1, PD-L1 inhibitors. Hyperprogression is a major concern with this type of immune checkpoint therapy as it results in reduced survival that is actually caused by the therapy when patients are treated with drugs such as Keytruda and Opdivo. Berberine. Berberine is a plant-derived extract that has been shown to lower blood sugar. Studies suggest in cancer that berberine may inhibit cell proliferation and promote apoptosis. There are other studies, however, that suggest berberine may have immune-suppressing effects, which does create concern in cancer treatment. But other studies have contradicted this finding, so berberine's role as a potential anti-cancer agent remains unclear. Berberine also seems to affect the microbiome, but it is unclear if the effect is beneficial in cancer. Karimi et al. Published a study in J. Acupunct Meridium Stud, April 2017, that showed in the mouse model that berberine caused a decrease in Treg cells in the spleen and also reduced IL-10, an immune-suppressing cytokine. Kim et al. Published a study in Cell Physio Biochem, January 31, 2018, that demonstrated that berberine suppressed breast cancer cell motility by decreasing TGF-B, a major immune suppressor. Selenium. As I had mentioned earlier in this book, I saw a poster presentation at the 2017 SITC by Lenicky et al. That described how supplementing with selenium could help patients who began failing PD-1 inhibitors to respond again. There is much in the literature describing the anti-cancer and cancer prevention properties of selenium. However, as I did further research, I discovered that there are few things to consider. The main supplemental forms of selenium is seleniumethionine, CMET, and occasionally selenium methyl L-selenocysteine. When I reviewed the literature, the more significant effects are seen with methylselenic acid, MSA. However, I have been unable to locate this in supplement form. Yen et al. Published a study titled Dietary Supplementation with Methylselenic Acid but non-selenomethionine Reduces Spontaneous Metastasis of Lewis Lung Carcinoma in Mice. It does seem there may be some conversion inside the body from CMET to MSA, but the studies I read still show that the results are inferior. So, for the time being, it seems selenomethionine may be the best option until maybe MSA becomes available. Lenicky et al. Published another study on oncoimmunology December 2016, modulation of MHC class I surface expression in B16F10 melanoma cells by methylselenic acid, MSA. They describe that MSA may increase MHC1 expression in tumor cells, which blocks a method that cancer can use to escape the immune response. In addition, MSA seems to have other immune-stimulating effects that decrease regulatory cells and many immune inhibitor substances like VEGF and PDGF. Curcumin. Curcumin is a plant-based compound that is a primary active ingredient in the spice turmeric. It has often been used as a supplement in cancer treatments. Bhattacharya et al. Published in J Biochem, June 2007, their findings that curcumin can protect tumor-mediated T-cell destruction. In addition to the direct effect on tumor growth, curcumin seems to increase tumor suppressor gene P53 and may enhance the anti-cancer immune response by decreasing TGF-B. EGCG, epigallocate-chingolate. EGCG is a polyphenol generally extracted from green tea. This is a supplement that is often used in cancer treatment. I would like to add a warning that there have been reports of liver damage at high doses. Therefore, EGCG should be used with caution. As with anything else discussed in this book, its use should be addressed with your doctor before taking it or engaging in any of these other therapies. Agawa et al. Published a study in Oncolet September 2012 demonstrating that EGCG may have immune-enhancing effects by inhibiting IDO, an enzyme we discussed previously that breaks down tryptophan, which is necessary for immune cell function. Studies have also suggested that the combination of EGCG and curcumin may decrease TGFB. In addition, several studies have shown that EGCG can inhibit cancer stem cells. Sulforaphane. Sulforaphane is obtained from cruciferous vegetables such as broccoli. It is necessary to have enzymatic production of sulforaphane, which is generated when myrosinase converts glucoraphanin into sulforaphane. So, when you are looking for this as a supplement, you will want the activated form. Castro et al. Published a study in CancerPrev Res, March 2019, that reported sulforaphane suppressed breast cancer stem cells. In regards to sulforaphane, there may be some contradicting aspects when it comes to the immune response. Some studies suggest it inhibits TGFB, which can reduce immune suppression in advanced cancers. A study by Joller et al., Cancer Immunotherapy, December 2016, showed that sulforaphane inhibits macrophage migration inhibitory factor MIF that can help cancer escape the immune system. Also, inhibiting MIF with sulforaphane can result in a decrease in MDSCs, which should further help the anti-cancer immune response. However, the authors, Liang et al., published a study in Ad Bioregul, January 2019, discussing how sulforaphane may be a double-edged sword when it comes to cancer immunotherapy. They state that sulforaphane acts pro-oxidatively in human T cells, which can inhibit their activation and tumor-killing functions. Certainly, it is hard to say if it is best to take the good with the bad in regards to sulforaphane. In general, I feel the benefits may overweight the negatives, but we certainly need more studies. Polydatin. Polydatin is a precursor of resveratrol. New studies, like the one published by Mealy et al., Cell Death Dis, May 2018, discuss how polydatin inhibits G6PD, a major glucose metabolism pathway. They showed that polydatin suppress cancer proliferation and metastasis. Though there is not a lot of direct links as to how this can affect the immune response, I did think it was at least worth mentioning. Remember that glucose metabolism results in significant lactic acid production, creating a metabolic immune checkpoint. Also, there is evidence that polydatin may decrease the WNT beta-catenin pathway, which in theory could result in increased immune cell infiltration in the tumor microenvironment. Clearly, more studies are needed, but the theoretical aspects of polydatin suggest that it may have affect anti-cancer properties and could result in further immune enhancement. Beta-glucans. Beta-glucans, also known as B-glucans, are polysaccharides that come from the cell walls of oats, barley, mushrooms, yeast, bacteria, and fungi. They bind to immune receptors and can attract immune cells into the tumor environment. Depending on where they are derived, B-glucans can have different effects. It is still unclear which source of B-glucans is superior, if any. Theoretically, the oat barley-based B-glucans may have an advantage, but that still is not fully proven. No matter the source, all types of B-glucans seem to have immune-enhancing properties. They are able to decrease regulatory cells and increase immune-stimulating cytokines, which can help turn up the heat in the tumor microenvironment. Since a cold tumor microenvironment is a major reason for immunotherapy failure, B-glucans have the potential to boost the response of immunotherapy. As I had mentioned before, the potential list of natural substances that may be immune-stimulating is exhaustive. I selected some of the ones that I thought had the most evidence and potential without being too overwhelming. A patient can only take so many supplements and medications. Now that you know about a few immune-enhancing supplements, we have to discuss some of the darker side of immunotherapy, adverse reactions. Thankfully, we can use intratumoral injection to minimize these side effects while maximizing success and reducing cost.